The overall goal of this study is to show how to implement automated procedures for culturing and differentiating human iPS cells into neuronal lineages as well as their automated imaging. Standardized automated procedures allow low experimental variation while ensuring high phenotypic reproducibility. In addition, the system can be adapted for the development of new protocols. Demonstrating the procedures will be Joachim Täger and Elisangela Bressan, postdocs from our lab.
In the graphic user interface of the automated system, click Resource Instrument Process View and select the Resource Instrument Process. Click Run Instrument Process and run the Run HEPA Hood and Reloading processes. Select the resources to be loaded, open the door, and load the cell culture plates and disposable tips in the appropriate positions as indicated by the pop-up images. Then decontaminate the door with 70% ethanol before closing and run the decontamination process. The system will be sterilized by UV radiation for 30 minutes. To load the plates with culture medium or dissociation reagent, execute the Run HEPA Hood step and open the door of the liquid handling station. After decontamination with 70% ethanol, place the reservoirs on the deck to the assigned position and delid them. Close the door of the